Okay, this is going to be a lecture about cellular respiration, probably done in a couple of installments. To start, we need to let you know what metabolism is. From your textbook, the glossary, it says it's the sum of all the chemical reactions that occur within a single cell or within all the cells of a multicellular organism. So what does that mean? We've already talked about chemical reactions. And those chemical reactions, we said, happen because of enzymes, right? Enzymes are going to speed up these chemical reactions to make sure that they're happening. And we said that enzymes can do chemical reactions two ways. We can either do that by making bonds, or we can do it by breaking bonds. And so metabolism is going to be either breaking down molecules into smaller pieces by breaking bonds, or we're going to build new big molecules by taking small stuff and putting bonds between them. We can take those two halves and we can say that the building molecule half of things is called anabolism. All we're doing is we're taking small things, putting them together into big things. Or the opposite, breaking down molecules, is called catabolism. You have two diagrams on your handout that show these two things. Anabolism, taking smaller parts, and with the use of energy, you're able to put those together by forming bonds. Where is that energy stored? That energy is stored in the bonds. Oops. Catabolism, on the other hand, you take those big molecules that are held together by bonds, and when you break them, although it does require some energy to break them, this is true, the energy that comes out of it is more than what goes in, so you end up with a net energy that comes out, and then you have these bigger molecules broken up into smaller ones. You had done a reading for me where we used the words endergonic and exergonic. Well, anabolism, where energy is put in and stored, these would be considered endergonic reactions. RXN is my abbreviation for reactions. And then during catabolism, when you break bonds to get a net energy out, these are called exergonic reactions. And so that just takes words from before and applies them to a new situation. The big thing here is that we finished talking about photosynthesis. And photosynthesis was taking carbon dioxide and water, doing something with it, and building these big glucose molecules. Well, glucose by itself is not a usable form of energy. What we actually need to do is we need to take that glucose molecule, we need to break it down, and we need to actually then recharge this energy molecule called ATP. Now ATP is called adenosine triphosphate. And so when you look at this picture of ATP, well, here's the adenine, the adenosine part. It's attached to a sugar, so the sugar plus adenine is called an adenosine. No big deal. Triphosphate means that you have 1, 2, 3 phosphate groups. What's a phosphate? It's a PO4 is what that's called. Okay? This is useful. If we look at it in a different way, here's my sugar, here's my adenine, here's my 3 phosphates hanging off. I'm going to take water, put it in there, put it in where? I'm going to put it in right here. And by putting water in there, I'm going to break it off. And by breaking off that energy, or breaking that bond, energy is released. And so now we have something called adenosine diphosphate, which just means that you've got this sugar and this adenine stuck together with 2 phosphates hanging off, this extra phosphate just kind of floating around. But the whole point is that by breaking this bond, you've got energy that's useful to do other stuff. You have a diagram where, I'm not quite sure why this is here. Let's get rid of that. You have a diagram on your page that has this ATP. I want you to put an arrow to this picture of it, and then another arrow to this picture of it. Because really this is the first step, the second step, the third step. Oops. The third step. We have ATP here with this high energy bond. The second step is water comes along, goes in, breaks this bond, and when that bond is broken, energy is released to be used somewhere else. Used somewhere else for what? In an endergonic reaction, because an endergonic reaction is one that needs energy in order to happen. Here is a little GIF file where we have our high energy bond. There's the water. Water then, poof, breaks the bond and energy is released. So it's go back to ATP, high energy bond. Water is present with an enzyme, and then it's going to break that bond and energy will be released. And so it just repeats over and over again. But here's the problem. Once you've broken that bond, how do you reform it? In order to reform it, because down here we're here, ATP was full, it got broken by water. Remember, water comes in, breaks it apart, and we get energy released to do something else. So then how is it we recharge it? Well, you can recharge it one of two ways. If you're a plant, you can recharge it using sunlight. And we saw that in the thylakoids. Sunlight was used to break water apart. But not only was it used to break water apart, it was also used to recharge ADP into ATP. You can go back and look at that flow chart. We had ADP coming into the thylakoid and we had ATP coming out. That was the recharging. That was just sticking this phosphate back on. Well, we're not plants. And so how are we able to do that? We as animals are able to get energy from the food we eat to recharge this ATP molecule. And that's what we're going to focus our attentions on. From your study guide, it says that I can explain how chemical energy is converted into useful cellular energy. Really? Basically saying, I can talk about how I use food as an animal to recharge, to stick this phosphate back on, to have this ATP molecule that can go somewhere else in the cell, give up its bond, break off that third ATP or that third phosphate, and let energy be released. Okay? And then how do we recharge? We have to recharge by having food available, and then we can transfer that energy somewhere else once we've done that. Some uses of ATP. Here you notice yellows being kicked out and greens being kicked in. But if you look, there are more greens on this side and more yellows on this side anyway. So we're actually working against nature. So in order to shove more yellows over and more greens in, you notice that an ATP molecule is used every time to force it to happen. And so remember, this isn't passive transport, it was the opposite one, which was active transport. And we usually call these protein pumps. It's a protein channel, just like we've seen before. But now this time we use energy to force things in the opposite direction that would seem normal. The second thing, which you've seen a picture of already, is anabolism. Building molecules from smaller molecules, building big from small. We need energy to do that. And that energy is in the form of ATP. Third one is just other random cell functions, but a big one, I will tell you, is muscle contraction. Muscles are nothing more than a bunch of proteins that slide over each other to contract. Well, when they slide over each other to shorten and contract, they're stuck there. The only way they can relax is if ATP is there, and that allows a muscle to relax so that it can contract again. So we've got ATP all over the place because we need it for lots of things. So when we look at cellular respiration, I want you to get rid of the idea of breathing, your lungs. This is not the lungs. This is what's happening in every single cell of your body. And what it is it's taking your glucose, and what it's doing, and this is kind of incorrect, food into ATP? Well, no, not really. What it's doing is it is going from glucose and recharging our ADP back into ATP. How are we going to have the energy to do that? We're going to break these bonds. And by breaking the bonds of glucose, we're able to transfer that energy over and be able to stick this third phosphate back on. So let's see how that happens. How and where? Where is where we're going to start first. Where does this happen? The first thing we do with glucose once it gets into the cell is in the cytoplasm, we rip the glucose in half. Rip it in half. Because remember, glucose is C6H12O6. And by ripping it in half, we end up with 2 3-carbon molecules. Okay, so we end up with carbon, 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 and a carbon, carbon, carbon. We rip it in half. By doing that, we call that glycolysis. Lysis means to break. Glyco means glucose. So it's the ripping of glucose. But you need to know this word. You need to know the word glycolysis. No oxygen is necessary to do glycolysis. And by just ripping a glucose in half, we do have some energy that is made. The energy that we're talking about is ATP, right? We're recharging ATPs. And when I say some, I really mean 2. 2 ATP are going to be recharged just by ripping glucose apart in half. And here's the picture of it. Here you start with your glucose. 2 ATP go in, so you have to put energy in to break something apart. That's true for anything. But look what happens as I rip off my hydrogens. That's what this is doing. It's that bus. There's another bus. We rip off hydrogens. When we rip off hydrogens, look what happens. We get 4 ATP. So what does that mean? If I had to put 2 in to break it in half, but then I get 4 out when I rip off the hydrogens, how many do I have net? I get 2 net ATP. And then these things, these pyruvic acids, well, their destiny depends on whether or not oxygen is present. I'm not quite sure why this happened again, but that's okay. So, this picture, glucose, goes through glycolysis, and I said that what happens to it depends on what is present. Notice that I have 2 different colors. I have glycolysis in blue, and then these words are also blue. And that goes with anaerobic respiration, which is one type of respiration. I also have glycolysis boxed in red, which goes with aerobic respiration, meaning that oxygen needs to be present. And where is oxygen? It's way over here. What is oxygen doing? Oxygen is there waiting for the hydrogens that are getting ripped off of my glucose. Because remember, glucose was C6H12O6. What are we doing? We rip off carbon dioxides, C's and O's. What do we have left? H's. Where do the H's go? The H's get caught by oxygen, and we end up with water as a product. Okay. What I want you to notice is that in glycolysis we get 2 ATP. In the Krebs cycle, we also get some ATP. But in this third portion, this electron transport chain, we get a whole lot more. And so between these two, we actually end up with 34 ATP, with 36 total. So anyway, let's move on. Based on this flowchart here, what do we need for cellular respiration, specifically aerobic respiration? We need a glucose to go in, and we need an oxygen to go in. What comes out? Carbon dioxide, water, and lots of ATP. So, what do we need to do aerobic respiration? These two. What do we get out of it? These three. ATP, water, and carbon dioxide. These groupings of molecules probably look pretty familiar to you. Because if I were to put this into an equation form, my reactants are going to be sugar, glucose, plus oxygen, yields carbon dioxide plus water plus energy in the form of ATP. Why does that look familiar? It should look familiar because it's photosynthesis backwards. Remember, photosynthesis was carbon dioxide and water with energy in the form of sunlight. Gives us glucose and oxygen. So really, they just get flipped. So aerobic respiration is really what happens to those glucoses that are ripped in half. They go into the mitochondrion. And in the mitochondrion, oxygen is going to be used. And when oxygen is used, lots of energy, remember, energy in the form of ATP, is made, and I'm going to say 34 net ATP are made just in the mitochondrion. When we did the chloroplast, I said that there was going to be, there was some anatomy that you were going to have to know, right? The stacks of pancakes were the thylakoids, and then you had the stroma was the filler. Well, in this case, we've got this inner membrane, which is folded, kind of like thylakoids were folded membranes on top of each other. And then we've got this matrix. This matrix is going to be the filler, kind of like the stroma was the filler. But by having these all folded inside the mitochondrion, what we've done is we've increased the surface area. And when you increase the surface area, and stuff is happening on this inner membrane, stuff is happening. When you fold that membrane, you get more of that membrane packed in. And if you have more of that packed in, we have more stuff happening. And so that's what's interesting about that. Okay, aerobic respiration. Remember I said there was a picture of a chloroplast that you had to love and cherish? Here's a picture of the mitochondrion that you have to love and cherish. Something I need to point out though, if this is the mitochondrion, what would this white space outside of the mitochondrion represent? It's going to represent the cytoplasm. Yes, cytoplasm. So what we have to remember is that glucose that comes into the cell and gets ripped in half, it's happening in the cytoplasm. Okay, so being ripped in half, remember we called that glycolysis. Glycolysis, what are we doing? We rip the glucose in half and we're going to send it into the mitochondrion. But we're ripping off H's, so we have to have another bus. So let's draw a bus. A bad bus. So we're going to rip off H's. We're going to put them on the bus. Okay? And the bus is going to go over here. Well, what are these called? This is called the Krebs cycle. And the Krebs cycle is taking those ripped glucose molecules, and those ripped glucose molecules are called pyruvic acids. Pyruvic acids. And really all they are is they're 3 carbon molecules. Because glucose was a 6 carbon, we rip it in half, now we have 3 carbons. What do? They come into the Krebs cycle. And the Krebs cycle is going to do 2 things. It's going to rip off more H's, so I'm going to draw another bus, another bad bus. Oops, that's a really bad bus. We're taking off the H's. That's what that's supposed to be. The H's are coming off the Krebs cycle. If we're bringing C's, H's, and O's into the Krebs cycle and we take off the H's, what's coming out? carbon dioxide is coming out. You need to know that carbon dioxide comes out of the Krebs cycle in addition to some ATP. Where do those H's go? Those H's come over here. What's waiting for it? Oxygen is waiting for those H's. And when the oxygen catches those H's, what comes out? Water. What do we call this process of oxygen catching the H's? We call it the electron transport chain. And it's from here that we get so many ATP. This is like 2 ATP. But you know what? It takes 2 ATP to carry this bus over. So really these kind of get canceled out. So what we say is, total we get 34 ATP that we get to keep. So 34 plus the 2 over here gives you how many from 1 glucose? 1 glucose allows us to recharge 36 ATP. Okay, so 1 glucose is going to recharge 36 ATP when oxygen is present. Okay. So here's just a cleaner version of it. This is the bus that I drew. This is another set of buses. Maybe you can say this is a single bus. Well, that's a double-decker bus. Whatever. Doesn't matter. I don't need you to know the names of these. I just need you to realize that hydrogens are being carried over to here. Here's a zoom in. The zoom in is, here's our 3 carbon. Remember we ripped it in half? Look what happens. We rip off a carbon dioxide. And we keep going. And we keep going. And what do? We rip off a carbon dioxide because we're ripping off an H. We rip off a carbon dioxide. And we rip off another H. Then we're over here, we're ripping off more H's and more H's. And so the Krebs cycle is taking those H's off and releasing carbon dioxide. Which is why I had you draw an arrow of carbon dioxide coming out of Krebs. It's very, very important. Because remember, glucose is C6H12O6. So what do? In Krebs cycle, we take the C's and the O's, release them, and where do the H's go? The H's go to the next section, the electron transport chain. There you go. Here's carrying H's. Here's carrying H's. What's happening to all those H's? All those H's are being dropped off. And what are we doing? Here's oxygen. Oxygen is catching those H's and turning it into water. And by doing that, that allows us to make all these crazy ATPs in the electron transport chain. So, the electron transport chain, remember, this is where oxygen is going. And that's where water is coming out. Here's just another visual. Carbon dioxide's coming out. Oxygen going in here and coming out as water. Here's Krebs and Calvin cycle side by side. And all it is in Krebs, carbon dioxide comes off, H's come off. In the Calvin cycle in the chloroplast, carbon dioxide goes in, H's go in, because you get sugar coming out of the other end. So they are opposite each other. Looking at the electron transport chains, here we've got, in the mitochondrion, we've got oxygens catching the hydrogens to make water, and we get lots of ATP as a result. With sunlight, remember we took that water and we broke it up, releasing the hydrogens. Well, we still get some ATP made. That's going to be the end of the first installment. I will come back in part 2 and talk about this diagram.